Students, welcome to biology class. We are dealing with the 5 kingdom classification proposed by R.H. SUDHAKAR, and in the last class we have already discussed about the 5 kingdoms. I have already discussed with you the 5 kingdoms: Monera, Protista, Fungi, Plantae and Animalia. And when I consider Monera, Monera is again been divided into 5 groups. We start with Eubacteria or true bacteria. In the last class, we have already discussed about the true bacteria. In addition to that, 4 other groups: Archaebacteria, Cyanobacteria, Mycoplasma and Actinomycetes are there, which we are going to discuss today. Archaebacteria, Cyanobacteria, Mycoplasma and Actinomycetes all are having the same characteristics of a prokaryote, and non-membranous membranous organelles are absent over there. They are not having any membranous organelle. Their nucleus does not have chromatin material, rather it is represented just by DNA. We can understand from this diagram about the Archaebacteria, which are very primitive in nature and very small in size. They are just having a plasma membrane, and the plasma membrane encloses the cytoplasm. Above the plasma membrane we have the cell wall, and inside the cytoplasm we have the DNA. Then we have ribosomes, and if we come to the characteristics of Archaebacteria, the Archaebacteria are most primitive, and it is said that these are originated at a time when there was extreme conditions in the earth. The temperature of the earth's surface was very high, and these are also known as the oldest of the living fossils. Those plants or organisms, plants, animals or organisms which were living once upon a time, which are found in fossil records and also found today, these are referred to as the living fossils. And Archaebacteria are found in the fossil records, also we find them today. So, we consider them to be the oldest of the living fossils. Their cell wall is made up of polysaccharides and proteins. These are categorized again to 3 groups: methanogens, halophiles and thermoacidophiles. If we come to the methanogens, you know they are related with the production of the methane gas. These are found in anaerobic conditions. They can grow in anaerobic condition and produce methane from carbon dioxide and hydrogen. These are found in marshlands and also in the gut of animals, and they are they have the major responsibility of production of the greenhouse gas methane. If we go to the second one, these are referred to as the halophiles. You know, halophiles are salt loving archaea, and they can grow in extreme salt condition. These develop a pigmented membrane, and taking energy from sunlight they can produce ATP, but they do not produce carbohydrate like the process of photosynthesis. If we go to the examples are Halobacterium, Halococcus, etc. Then comes the thermoacidophiles. The name comes from high temperature and also acid producing ones. These we find in hot sulfur springs and the temperature of these springs may be up to 80 degrees. These organisms oxidize sulfur to produce sulfuric acid. The examples are thermoplasma and pyrococcus. Then we come to the next one, cyanobacterium. These are also referred to as blue-green algae or mixophyceae or cyanophyceae, now known as cyanobacterium. These are prokaryotic and found almost everywhere in the world under all conditions. This can grow wherever minimum requirement of the plants are there, like there is water, there is sunlight, there is presence of gases and some nutrition. These are also found in symbiotic association with different groups of plants. Cyanobacteria have a specialized structure or a cell which is referred to as heterocyst. With the help of the heterocyst, they can fix atmospheric nitrogen. That is, these are unicellular, but these may be found in colonial or filamentous form. These have these heterocysts using which they can fix atmospheric nitrogen. As they are capable of fixing atmospheric nitrogen, they can grow anywhere where there is availability of water or moisture as well as sunlight. These are important in ecological point of view because they are the pioneers in water bodies when there is a water body without a living organism, and they promote succession by enriching the water body with nutrients, because these are the nitrogen fixers. These are used as food and also as fodders. Then we shall come to the next one, that is the mycoplasma. Mycoplasma are the smallest prokaryotes without cell wall. These infect a large number of animals including man, and this also causes infection to plants. Their size is very, very small, that ranges from 30 nm, 300 nm to 20 nm in their dia. They are unicellular and also smaller than the viruses. Their protoplasm contains both RNA and DNA. Their protoplasm contains both RNA as well as DNA. This reproduce by a process similar to binary fission. If you go to the sexual reproduction, there is no sexual reproduction in this group Monera like the prokaryotes, and we consider this to be intermediate between bacteria and viruses, and these are causing a lot of plant diseases. I have already mentioned that this causes many animal diseases from where it is actually identified, and they also cause diseases like aster yellow, little leaf of brinjal, etc. Diseases like rheumatism, arthritis and other respiratory diseases are also caused by mycoplasma. Examples of mycoplasma are Mycoplasma hominis and Mycoplasma fermentans. We can see here the mycoplasma has no cell wall, and there are soluble RNA, ribosomes of prokaryotic type, and then the DNA is a double helical structure, but not circular. Then we shall come to the next group, actinomycetes. Actinomycetes are filamentous bacteria, aerobic, and they grow abundantly in soil, water, mud, manure, and milk. These appear like filamentous fungi. These produce extracellular enzymes which enable them to utilize polysaccharides, proteins, and their organic substances present in the soil. These are mostly saprobes. They live upon already synthesized organic matter. Some of them are also parasitic. Frankia is a species which is known as a nitrogen fixer. They reproduce by conidiospores, and they can bring about fermentation. These are source of some important antibiotics like streptomyces, actinomyces, etc. With this, actually we have completed the salient features of the group Monera. Then we shall come to the salient features of Protista. We have already discussed the protists are unicellular, eukaryotic, colonial or filamentous, but not organized into tissues. The basic difference between the protists and the Monera are the Monera are the prokaryotic ones, whereas the protists are eukaryotic ones. The prokaryotic cells, we have already discussed that the prokaryotic cells do not have a nucleus. So, this is a eukaryotic cell and this is just a prokaryotic cell where we shall see the DNA is a double stranded circular DNA molecule. There is no association of histone protein or any other protein with this DNA. They remain in the clearer zone to in the cytoplasm, more or less in the central region, and that is referred to as the nucleoid. Whereas in prokaryotic cell, we shall see the nucleus is well developed and the nucleus is covered by nuclear membrane. It is enveloped by two membranes. The nuclear membrane is having nuclear pore, and inside that we have the genetic material DNA remaining associated with histone and non-histone proteins. The histone proteins are 5 different types. The histone proteins are 5 different types like H2A, H2B, H1, H2A, H2B, H3 and H4. The long DNA molecule is packed with these 5 different types of histone proteins, and they form the chromatin structure. This in detail you will be discussing during your cytology studies. Well, the next important point between the Monera and the Protista, here you have membrane bound organelles, the endomembrane system, you have chloroplast, you have mitochondria, etc. Here you will have membrane bound organelles, and here the ribosomes are ATS type, and here you have ribosomes which are 40S type. And with this actually we shall go to the protista. We shall discuss about the second group of organisms coming under protista. Protists are unicellular eukaryotes, colonial or filamentous, but not organized into tissues. So, here we shall see that the Monera is having prokaryotic cells no true nucleus is there, but in case of protists we have true nucleus. If we make a comparison between the prokaryotic cell and eukaryotic cell, we shall see in the prokaryotic cell there is no nuclear membrane or nuclear envelope, there is just a double stranded circular DNA molecule, and this DNA molecule is without any histone or non-histone proteins. But in case of eukaryotic cell, we have a well developed nucleus. The nucleus is having nuclear envelope, and the nuclear envelope is made up of two membranes, the outer nuclear membrane and inner nuclear membrane, and there are endomembrane system made up of many membrane bound organelles like this. So, we shall have mitochondria, we shall have chloroplasts, and we shall have also ATS type of ribosomes. In this case, in case of prokaryotes, we shall have 70S type of ribosomes. In this case, DNA is associated with five different types of histone proteins. H1, H2A, H2B, H3 and H4. Well, these five different histone proteins, they together make the nucleosomes and then the chromatin material. This is the specialization of eukaryotic cells. Now, we shall see this group includes algae, protozoa and euglenoids, and all the cells are having cell wall made up of cellulose. In the previous case, we have seen in case of Monera, there are different elements that make the cell wall, but here when there is cell wall, these are made up of cellulose, and we have three groups under protists we shall be studying. this protista algae, then the protozoa and the euglenoids. This is not the general algae, but we shall refer to as protista algae. Slime molds without cell wall, we shall find slime molds under this, and this is interesting to see that slime mold will not have cell wall during its vegetative phase, but will have cell walls developed during the reproductive phase. Then we shall go to the next characteristics. DNA associated with histones and chromatin is found. There is presence of cilia, flagella and pseudopodia with which they move. These are the locomotory organisms, and we shall find all type of nutrition over here. The nutrition may be autotrophic when they synthesize their own food. So, the algae is having chlorophyll and accessory pigments, using which actually they bring about photosynthesis, and they are autotrophic in nature. Then they are also saprophytic when they depend on already synthesized organic matter, and they are also parasitic. When they are parasitic, they live on living organisms and cause diseases to the hosts. We shall then come to the major groups of these proteins. The first one is photosynthetic proteins, or also we say them to be the protista algae. These make the phytoplanktons. Phyto refers to plant and planktons refers to the free floating or freely swimming organisms. They carry out about 80% of photosynthesis in the earth, and there are three groups of protista algae. These are dinoflagellates, diatoms and euglena like flagellates. The second type of the proteins are the consumer decomposer proteins, or we say them to be slime mold. The third type of the proteins we shall say as the protozoan proteins, and let us come to the photosynthetic proteins. These are also known as protista algae or phytoplanktons. Some of these are motile and some are non-motile. These are divided into three groups: dinoflagellates, where we shall find these made up of single cell. These are motile, sometimes non-motile. They show amoeboid, palmelloid or filamentous habit. The body is enclosed by a rigid coat called theca or lorica, made up of two or more plates of cellulose and pectin, variously sculptured. These walls are variously sculptured and they differ from each other. Each motile cell is having a pair of flagella. The main pigments are chlorophyll A, C, carotenoid and xanthophylls. These are mostly photosynthetic, sometimes parasitic, symbiotic or holozoic. Dinoflagellates reproduce mainly by asexual method of reproduction. However, few species show sexual method of reproduction. The examples of these are Gymnodinium, Glenodinium, Dinophysis, etc. Then we shall come to the next ones, the diatoms. This is the largest group of protists. There are about 200 genera and 10,000 species belonging to the class Bacillariophyceae. Cells show bilateral or radial symmetry. This is a very peculiar character of this. They will have a symmetry of their body. These are mostly unicellular, some are filamentous and some shows colonial habit. Cells of different shape without flagella surrounded by a rigid siliceous cell called the frustrule. Cytoplasm surrounds a large central vacuole and a single nucleus. Pigments are arranged in chromatophores. Golden brown in color, and they reproduce both by vegetative, asexual as well as sexual way. Then we come to the third one, the euglena-like flagellates. These are also referred to as euglenoids. These are freshwater organisms. They show both plant and animal like characters. These are unicellular and flagellated, found in places where there is fresh water like ponds, ditches and also in slow running streams. These are without cell wall and at the anterior end of the body contains a cyphostome, and locomotion is by a long whip like flagellum. Interestingly, this is without cell wall. You can see it from the diagram, the characteristics of this. This contains a number of chloroplasts and chromatophores. A large contractile vacuole is present, and the function of the contractile vacuole is osmoregulation. Nutrition is autotrophic or holophytic, sometimes it is saprophytic. They reproduce asexually by binary fission. There is also formation of cyst and pamela stage. Then we shall discuss about the consumer decomposer protease. These are also referred to as slime molds or protistan fungi. They have an amoeboid body, a vegetative phase without cell wall, but later on during reproductive phase there may be formation of cell wall. These are phagotrophic in nature. They reproduce by asexual as well as sexual methods. These are of two different types, plasmodial slime molds and cellular slime molds. Well, with this we finish this protease. We shall now go to the next group, fungi. The singular of this we read as fungus, Fungus is achlorophylous. These are without chlorophyll and other pigments which are responsible for photosynthesis in plants. So, these are plants, but which are not autotrophic in nature. These are eukaryotic, spore bearing, filamentous, branched and have a cell wall made up of chitin found everywhere in the earth. So, we have seen different types of material that make the cell wall of different groups, like in Monera mostly we have seen these are made up of murines or peptidoglycan. In some other cases we shall see these are made up of carbohydrates and proteins like in archaea. In algae we have seen these are made up of cellulose, and now in fungi the cell wall is made up of chitin. The thallus is called mycelium. The mycelium is made up of slender filamentous hyphae. These are heterotrophic, they may be saprophytic, living on dead or decomposed organic matter, or parasitic, they live upon the host bodies deriving nutrition from them, and some of these are also having a symbiotic association with algae. Some algae in a symbiotic relationship with the fungi form a life form referred to as lichens, and lichens are also pioneers. They can survive in very tough life conditions. When there is succession or starting of life process in a rock surface, we shall see the pioneers are lichens. In this case, the algae produce food material and the fungi gives protection and water to them. There is another group of fungi which grow in association with roots of higher plants as well as gymnosperms, and they are referred to as mycorrhizae. Mycorrhizae are responsible for nitrogen fixation. There is great variation in the shape and structure among the members of fungi. There is great variation in their thallus structure. This may vary from a unicellular or single celled, uni or multinucleated thallus with long slender filaments. Saccharomyces cerevisiae, the common yeast, is a unicellular form, and we see many of the fungi are normal, having the hyphae making the mycelium. Some of the hyphae are having septation walls or septum separating different cells inside the filament. Some are aseptate. In some cases, there will be repeated mitotic division resulting in the formation of many nuclei, but there will be no septation that results in the formation of a sinusotic hyphae. Reproduction in fungi is by all the methods of reproduction like vegetative, asexual, and sexual method. Vegetative reproduction in fungi is again by fragmentation. When the mycelium comprising of hyphae are been fragmented, each fragment under favorable condition can grow into individual plants. Then fission under favorable condition, when the nutrition supply and supply of food and all environmental conditions are favorable, fungi grow very fast by just fission, whereby the genetic material will be divided into two and there will be laying down of a wall resulting in formation of two cells out of one cell. There is also budding, formation of a bud like we see in case of Saccharomyces or yeast. There will be formation of buds and the bud will be cut off from the mother cell and the bud will develop into individual cells. Then we shall come to the asexual reproduction in fungi, and asexual reproduction takes place by 5 different types of spores like zoospores, conidium, oidium, chlamydospores, aplanospores, etc. In case of zoospores, the spores produced will have flagella with which they can move. So, that is why these are referred to as the zoospores. Conidia, plural of conidium is conidia. Conidia develop on conidiophores on the margin of the apex of these some of the fungi, and there we shall see chains of conidia are formed. Each conidia, when get suitable environmental condition, they can grow into individual plants. Then oidium is also a spore using which they can germinate to individual fungus under favorable conditions. In chlamydospores, there are no flagella and they are thick-walled oval shaped ones, and aplanospores will have some thick walls surrounding them. Sexual reproduction includes fusion of two compatible nuclei. This fungi shows two different types of conditions. Inside the cell, we shall have one type of genetic material, and in some other cases we shall have different nuclei which we refer to as heterokaryons. Some fungi show male and female sex organs. Then let us come to the classification of these fungi. Fungi are classified into 4 different classes based on morphology of mycelium, asexual spores, their color, shape, etc. On the basis of this, these fungi are classified to Zygomycetes, Ascomycetes, Basidiomycetes and Deuteromycetes. We shall see in case of Zygomycetes, members are aquatic and they live on decaying food in moist and damp places. Mycelium is septated and cynocytic. Asexual reproduction is by zoospores or aplanospores. Sexual reproduction may be isogamy or anisogamy, or also oogamy is seen. This results in the formation of zygospores. These zygospores undergo meiotic divisions and form the 4 cells. These individual cells will now grow into independent plants. The zygotic meiosis results in bringing down of the diploid characteristic of the zygospore to again the haploid gametophytic plants. Examples are rhizopus, mucor, albugo, etc. We have heard about the black fungus disease that has caused a lot of deaths. This infection has been found after infection with the coronavirus, and this simple zygomycetes mucor is causing that. We can see the structure, very simple structure from the diagram which has been provided in this slide. Then comes the ascomycetes. Ascomycetes word is derived from an ascus, which is a sac or bag like structure, and for this fungus is referred to as sac fungus or ascomycetes. This includes the higher fungus or higher fungi. Thallus is uni or multicellular. When multicellular, they have septation of the hyphae, and this may be also branched, and the wall of this fungus is made up of chitin. Asexual reproduction is very common, is through spores called as conidium. We shall have formation of the conidiophores, and chains of conidia are born one above the other, forming in quite a number of conidia, and when the conidia are dispersed and they get suitable environmental condition, they grow into independent plants. Sexual reproduction occurs by sexual spores, and these are forming ascospores. In one ascus, there is formation of 4 to 8 haploid ascospores. Examples of the ascomycetes are Saccharomyces, the yeast, Penicillium, Aspergillus, Claviceps, etc. These may be saprophytic. These are also found to cause diseases. So, we can see from the diagram there is formation of 8 ascospores within the ascus. Next, we shall study about the basidiomycetes. These are also referred to as club fungi, and commonly we say as mushrooms. Basidiomycetes grow on soil, logs, tree stumps, and also on living plant bodies. These are the most advanced fungi and are the best decomposers of food. Mycelium is branched, aseptate, there is no septation. Sometimes we shall find Doliper septum, a septum having a pore inside that, and sometimes we shall have septum with clump connection. Primary mycelium is monokaryotic phase, where we shall have single nucleus and is haploid and short lived. The secondary mycelium is dikaryotic, where we shall find two different type of nuclei in this, and in this phase we shall find the two nuclei having genetically different strain, and then these are very long lived. So, the one which ferinates actually is the dikaryotic phase. Asexual spores are not found. There is vegetative reproduction by fragmentation. Sex organs are not found. Sexual reproduction involves plasmogamy, karyogamy and meiosis. So, what we understand by plasmogamy and karyogamy, we shall see the fungal cell. Suppose we see this is the nucleus, this is the cytoplasm. When there is fusion of nuclei of two cells, these two nuclei fuse, this is referred to as karyogamy. And fusion of the cytoplasm of the two cells is referred to as plasmogamy. So, if we start with a unicellular fungus, we find a uninucleated one, and then there is a compatible one. If there is fusion between these two cells, there will be fusion of the nucleus and fusion of the cytoplasm, and after that actually the nucleus become diploid, and when this diploid nucleus has to come back to the haploid one, then there has to be a meiosis or a reduction division, by which actually we shall have formation of 4 cells, 2 having one type of nucleus and the other 2 have other type of nucleus. If it is a heterokaryon, if this carries both these type of nuclei, a positive strain and a negative strain, that will become a heterokaryon. We see here there is no sex organ, that is interesting, and in course of evolution from the Zygomycetes up to the fungi imperfecti that we shall read now, we shall see there is degeneration of sex, gradual degeneration of sex we shall find in case of fungi. So, with more advanced fungi, we shall see the sex organs are gradually reduced. Agaricus, the edible fungi, and there are many disease causing fungi also like Puccinia belongs to etcetera belongs to Basidiomycetes. We can see the diagram of a mushroom that belongs to Basidiomycetes. Then we shall come to the last group, the Deuteromycetes. Deuteromycetes is also known as fungi imperfecti, where perfect sexual stages are either absent or not known. We have so far not found the perfect sexual stages with this group of fungi. They reproduce only asexually by formation of conidia or conidiophores. The mycelium is septate or also they are branched. Most of the Deuteromycetes are decomposers, while some are saprobes or parasitic. We can see Alternaria solani is a member of Deuteromycetes that cause early blight of potato. It is a very disastrous disease of potato. Coletotrichum fulcatum causes red rot of sugarcane, that is a pain to the sugarcane farmers. Cercospora personata that causes tica disease of groundnut, and Helminthosporium oryzae is resulting in leaf spot of rice. Now, we shall go to the economic importance of fungi. Fungi are used as food like the mushroom. There are also mushroom which are used as medicine. Many of the mushrooms causes poisoning also. Fungi are used in industries for production of antibiotics. We know most of the antibiotics are produced from fungi. Organic acids, vitamins, enzymes and other bioactive molecules are derived from fungi in various industrial processes. We know yeast is the fungus that is used in fermentation and production of alcohol. These are also used in biotechnology. Many of these, mostly the yeast, are been biotechnologically modified to yield specific products in bioreactors. These are responsible also for many plant and animal diseases. Aspergillosis in man is a very bad disease. The candidal disease, the common ringworm, dandruff, etc. are caused by this fungus. These are responsible for destruction of timber and timber products. These are responsible for spoilage of food and mushroom poisoning, etc. This, as a symbiont of the lichen, is a pioneer in evolution of the ecosystems or ecological succession. As a succession pioneer, is a decomposer, and for its many industrial uses, this fungus is considered to be economically important for the society. Well, students, we have covered these 5 kingdom classifications fungi In the next class onwards, we shall be discussing about the other groups, Plantae and Animalia. Thank you. See you again in the next class.